So the final segment is very exciting, and I think especially this year and this new therapy session includes one where I think that it's probably had the biggest buzz of ASH has been in this segment. So I'm going to show you some of what we've heard here in Atlanta and get your personal feedback about some of these results, okay? So I'm going to start with what we've heard about before. I just draw attention to the fact you guys have been involved with these studies using venetoclax, the BCL2 inhibitor, monotherapy, very interesting activity in 1114 related to the BCL2 level. Published in Blood just November 30th, and then the combination data combining with bortezomib, activity especially in the bortezomib naive, but also in previous exposure, again just published in Blood. Very, very interesting sort of precision approach. Then the trials are continuing with selinexor, and we have the STORM trial moving forward. I think that a little bit more challenging, I would say personally, related to toxicity, but really, really important to have one more agent that is active in this very refractory quad, penta-refractory situation, so very important. Then we do have the other antibody, the izatuximab, and I'll ask you about this, Joe. This is you. Actually, both of you were part of this. Both of you. So I think we mustn't forget that there is actually another anti-CD38 coming along, and then of course the abstract which probably attracted the biggest buzz at the meeting, which is the anti-BCMA CAR T-cell therapy, the so-called Bluebird trial, the BB2121 there. So I draw attention, a small study, 21 patients, and CRs at 56%, VGPR 89%. So obviously deep responses in very, very heavily pre-treated patients. Up here you see this. But I would also draw attention to this down here. You know, not all of these remissions are being sustained. Certainly there are some MRD levels being achieved, but I think that we've talked about MRD. One needs to look at that carefully. What is the level of the MRD? Is it sustained? And I think balancing that efficacy, what about the toxicity, the cytokine release? Quite challenging. Most of the CRS was grade 1 and 2, but certainly grade 3, got to be very cautious about that. And then the immediately following abstract, abstract 741, was the anti-BCMA monoclonal antibody with the drug conjugate. Again, very, very interesting results. This was an expansion of a study presented last year, actually. 60% response rate, but with deep responses, stringent CR, 15 VGPRs, so rather well tolerated, an unusual corneal toxicity, which apparently is quite manageable, but needs to be watched. And so very, very interesting to see these two approaches to the same target. And so finally, I just want to say that we have all of this different information in these different classes, the monoclonal antibodies, the immune modulatory agents, the proteasome inhibitors, and these other classes of drugs. Very, very fortunate to have all of those. And so my question to both of you, and maybe Paul, you would like to start first on this one. What do you feel is the most promising approach right now?
Well, you know, I was asked to do a sort of best of ASH for a large patient website, and it was, of course, the CAR T platform. I thought the results were remarkable that Hez has presented. I think, again, you know, an overall response rate of 94%, a CR rate north of 50%, and the VGPRs, as you showed so nicely. So really impressive. I think the Flu-Cy platform does appear to matter because Adam Cohen presented a very nice presentation, very thoughtful presentation from the UPenn group the day before, where 4 of 9 patients responded with only Cytoxan. So what this tells you, I think, about CAR T...
But I think the toxicities were a little challenging in that particular study, as I recall.
Exactly. But what it tells you, and I couldn't agree with you more, Brian, but my point is what it tells you is that this is a highly nuanced approach, and that subtle changes may make big differences. So there's a lot to learn. And obviously we have the strength of the Chinese data, which I think is fascinating.
Yes, the Shanghai
and they're
team,
using this two antigen approaches, which has been really impressive.
So it's interesting to think of these in terms of both efficacy and toxicity.
Absolutely. And that's my point, is that it's highly nuanced. And I think that CAR-T is fantastic. I mean, it's clearly one of the most interesting and promising new approaches. But I think we have to be careful because it's early, and it's essentially, when you look at these experiences, they are essentially a numerator-based experience, because we were part of the Bluebird trial, and you have, a waiting list that's almost a year long to get onto these studies. So there's selection in that process.
Absolutely.
Irrespective of that, the data are very, very impressive. I think the CRS is relevant. We have to remember in myeloma patients, because the allo experience tells us this, our patients are vulnerable to immunotherapeutic toxicity. And the best example of that is what happened with checkpoint inhibition and IMiDs in the Pembro studies.
So
we have to be very aware of great efficacy, but let's be also careful about side effects.
Right. So
and that's relevant.
Very, very important. And so Joe, what's your take on this? The CAR-T and then the antibody?
So much to say, but very briefly, I think those first few molecules you noted, the venetoclax, it's, if you will, a boutique drug to some degree, at least now by itself.
Very helpful. Very helpful.
We've been able to use it off study in a number of patients with translocation level 14. It's a pill, very well tolerated. Some of our patients with very aggressive disease just seem to immediately respond to this drug. Selinexor, I agree with you. It has tremendous challenges from a GI toxicity, but if we can get people through that first month, often it's much more tolerable. I do think isatuximab will have a role I think the more the merrier in some respect. That's why we work on it together. We have to.
The PI
daratumumab has been so prolific. It won't have that same effect, of course. And now we're working with an even more rapid infusion of the drug, where it can be given over 75 minutes.
Right.
My quick take on the CAR T-cell therapy is very similar to what Paul has said. We're all excited about it.
Yes.
But we're still got some early model CARs here. I mean, we really need to work out a lot of the bugs. Some of these are a little bit clunky. They're not quite efficient yet. I don't say that to be disparaging. I just mean that not all CAR T-cell therapy is the same. And we learned from our leukemia and lymphoma colleagues, it took them a number of years to refine this. And although, as you say, waiting lists are long, I think the CAR T-cell therapy that we're going to have at a year and 2 years from now will be that much better, where it will be more efficacious and it'll be safer. I am a little nervous at the number of people that have relapsed fairly soon thereafter. So this idea that people think it is a cure, or that people think it's just a be-all and end-all, we have to be cautious. It's like any other treatment we've had, albeit very promising in very relapsed disease.
So this last point about where patients are relapsing, and I would balance that against what will undoubtedly be an enormous cost. And so with the approval of the CAR-T in the childhood leukemia setting, this raised that issue, and the number that started to float around immediately was several $100,000. Between that and $1 million, once you consider all the logistics and you reach a high commercial level of this preparation, which is by no means trivial.
And that speaks to access too.
Yes.
So it's not just the pure cost, it's how many people are going to have it access. I mean, looking way down the line, we'd love an off-the-shelf like CAR T-cell approach, where it doesn't have to go through all the preparation, but we're clearly not there yet.
That's a little disappointing, actually, initially, right?
Very much so. So time will tell.
Yeah, and if I may, I'll pick up on that point, Joe, because I think it's an excellent one you make, the question of being off the shelf. And that's what I really like about the BCMA antibody drug conjugate study that Suzanne presented. And again, in full transparency, I'm obviously a co-investigator there. But the fact is, what we saw with that antibody, every 3-week infusion, it's a 1 to 2 hour infusion. It's a walk in the park by comparison. The corneal toxicity, to your point, Brian, is very manageable.
Yes.
You've got to have a good ophthalmologist, but it's straightforward. We saw thrombocytopenia, but really beyond that, not much else. And I think that kind of striking efficacy from a monotherapy, and it's worth noting in Suzanne's presentation that responses were seen in 40% of patients who'd had prior DARA. And I think that points to the success of the future. And one last closing comment, I completely agree with you, Joe, that CAR T is great, but what we mustn't do is think of this as a sort of zero-sum game. And there were some very nice presentations of additional drugs where we're seeing salvage in the setting of relapsed refractory disease. And a variety, touch on the selinexor, which I think is absolutely correct. Fascinatingly, a straightforward modified alkylator called melflufen. Mary V presented lovely data in the Horizon trial.
So a lot more to learn in this setting. I would have to say I found the results with the GSK monoclonal antibody intriguing. Deep responses, well tolerated.
I agree.
And so I think that we need to continue, watch this space. Watch this space and see what emerges. But really tremendous hope. And excitement, hope for all patients who might be watching. A lot in the pipeline, a lot very close to being available.